In this video tutorial, we will study about the topic DNA sequencing. DNA sequencing is the process of working out the order of building blocks of bases in a strand of DNA. Before we can sequence the DNA, it has to be cut into smaller pieces. Now, it inserted into the plasmid DNA and then transported to bacterial cell. This makes possible to produce lots and lots of copies of it as bacterial cells multiplies. The DNA is now isolated from bacteria and sent for sequencing. The isolated DNA is now transferred to a plate where the sequencing reaction takes place. A mixture of ingredients is added. This includes 3 DNA bases A, C, G and T, DNA polymerase enzymes, DNA primer and modified DNA bases labeled with colored fluorescence tags are also added in it that called terminator bases. To start the sequencing reaction, everything is heated to the temperature 96 degrees Celsius. This separates the DNA into a single strand. The temperature is then lowered to 50 degrees Celsius. This enables the DNA primer to bind to the plasmid DNA. The temperature is then increased to 60 degrees Celsius and the enzyme DNA polymerase binds to the primer DNA. DNA polymerase start making a new strand of DNA by adding unlabeled DNA bases to the target DNA. It continuously adds DNA bases until a terminator base is added. These terminator bases have been chemically altered so that no more bases can be added to the new strand of DNA. Once a terminator base is added, the DNA polymerase enzyme stops making DNA and falls away from the strand. Everything is heated to 96 degrees Celsius again to separate the new DNA strand from the original strand. This process of heating and cooling is repeated again and again to produce lots of fragments of DNA of different lengths. The length of DNA fragments depends on when the terminator bases got added. To read the sequences of DNA, the various fragments are separated by length using a process called electrophoresis. A capillary tube is lowered into each wall of the plate and the electrical charges is applied. This causes the negatively charged DNA molecules to move through the capillary tube. Each capillary contains a porous gel from which the shorter fragments of DNA move through easily than the longer DNA fragments. As a result, the fragments are arranged by size from the shortest to the longest. As the DNA fragments come to the end of capillary, a laser makes the terminator bases light up. The color is detected by a camera and recorded. Each terminator base is labeled with a different color: A fluorescence green, C blue, G yellow and T red. The shortest DNA fragment will read first and the longest read at last. The sequence machine records the color of the terminator bases as a series of colored blocks. Each colored block represents the labeled terminator base and of each DNA. By converting the colors into letters, we get the sequence of our piece of DNA. Thanks for watching.